All right, in this video, we're going to talk about patterns of life, characteristics of life, the study of life. So, we're going to look at the characteristics that identify living things as living things. What are the traits that all living things have? We're going to look at cross cutting concepts in biology. And we're going to look at how fields of biology differ in their approach to the study of living things. Also, going to talk about the importance of the metric system in biology and science in general. So, biology is the study of living or once living things. All living things have certain common characteristics. They're things that are found in all living things. Living things share these basic characteristics. Number one, they're made of cells. This is one of the reasons why most scientists would say that viruses are not living things, because they don't have cells. They reproduce. They make more living things using cell division. So there's two kinds of reproduction. There's sexual reproduction. It involves two parents combining cells and combining genetic information to produce offspring that are not identical to either parent. Most multicellular living things use sexual reproduction. Asexual reproduction is where a single parent divides to produce offspring that are identical to the parent, basically clones. Most single-celled living things reproduce this way. All living things have a nearly universal genetic code. The DNA sequence that codes for a particular protein or the particular amino acid is the same in all living things, at least almost all living things. There are a few exceptions to that, and we'll talk about that when we get into genetics. They grow and they develop. So growing is simply increasing in size, but not changing the structure and function. Development are the changes that occur to a living thing as it matures. In some cases, that's very simple. In other cases, it's pretty dramatic, where you have caterpillars changing into butterflies, tadpoles changing into frogs through metamorphosis. That's a much more dramatic development. If you look at from an early embryo to an adult mammal, including humans, there's big differences in those different stages of development. They respond to changes in their environment. And some animals might migrate when the temperature changes. Some animals, their fur color changes from summer to winter to blend in with the environment better. Lots of things, and we'll talk more about that. So a stimulus is a signal from the environment that a living thing responds to. They maintain an internal balance. We call this homeostasis. They keep their internal conditions within a set range. If they're out of balance, that means there's something going wrong. They have a disease or an illness. So homeostasis is the tendency of living things to maintain their internal environment different from their internal environment within the set normal range. They change over time. This is evolution. Living things today look different than living things did in the past. Changes in the population of species over time. So cross-cutting concepts in biology. One of them is cause and effect. Mechanisms and explanations. So again, there's a change. It causes an effect in a living thing. Systems and system models. Stability and change. Patterns. We see lots of patterns in the study of biology. Scale, proportion, and quantity. Energy and matter. Talk about energy flowing through ecosystems. We talk about matter cycling, and then conservation. Structure and function. Again, structure and function always go together. What something looks like is related to what it does. So fields of biology, and there's lots of them, from things like studying microbiology, studying genetics, inheritance, studying ecology, and depending on what they study, what level of organization they're studying at, they use different tools and different techniques and different procedures to do that. Ecologist doesn't do a lot of controlled experiments, whereas a microbiologist can do much more controlled experiments in a laboratory setting. So in science, we have to measure things oftentimes in our experiments. So we've come up with a system of measurement that is agreed upon by scientists around the world. That way, no matter where somebody's studying a particular thing, the units of measurement are the same, and we can relate that from one person to another, one place to another. We call these SI units, and they were developed many years ago. Scientists got together and decided this is what we're going to use to measure these characteristics or these traits. Most SI units are metric measurements. Not all of them, but a big part of them are. So common metric measurements that are part of the SI system are, for length, we use the meter. And then we have prefixes in the metric system that tell us, whether that's a fraction of that or it's a multiple of that. Like a centimeter is 1/100th of a meter. It takes 100 centimeters to equal a meter. A millimeter is 1/1000th of a meter, 1,000 millimeters in a meter. A kilometer is 1,000 meters. It's 1,000 times bigger than a meter. With mass, we use kilograms, and you can see that. Volume, we typically use liters, but milliliters and cubic centimeters are common terms we see in measuring volume, and they are equivalent to one another. Temperature, we typically use the metric system, even though that's not the technical Kelvin, or the technical SI unit is Kelvin, which is based on absolute zero. The metric system, water freezes at 0 and it boils at 100, which is different from what we're typically used to using the Fahrenheit system. So here's some metric conversions. If you wanted to convert 0.45 liters into milliliters, we would have to multiply that by 1,000, and we would get 450. We move the decimal place 3 places to the right. Going from kilometers to meters, kilo means 1,000, so again we would multiply by 1,000. Milligrams to grams, we would divide by 1,000 because milli means 1/1000th, so that would be 5 grams. And you can see some of these others here.